Good morning, students. Now coming to bacterial reproduction. Bacterial cells are very minute organisms and are show great diversity. And also, bacteria they are present in every habitat. So, bacterial cells are omnipresent, means in worldwide distribution. This is also called ubiquitous, means present in each and every way. At the same time, bacterial cells also reproduce by two methods. One is binary fission, second one endospore formation. Means there are two methods in reproduction of bacterial cells. One is asexual reproduction, second one is sexual reproduction. So, bacterial reproduction is of two types. One is asexual reproduction. One asexual reproduction. Okay, and second one sexual reproduction. So, there are two methods. Bacteria reproduce by two methods. One is called asexual reproduction. Second one is called sexual reproduction. So, sexual reproduction is not true sexual reproduction because there is no the production of male and female gametes, no fusion of gametes, and there is no the formation of zygote. But genetic material is transferred between two bacterial cells. So, it is not a true sexual reproduction. At the same time, asexual reproduction, it does not include the formation of male and female gametes, and there is no the fusion of gametes, and such type of reproduction method is considered as asexual reproduction. So, bacterial cells they reproduce asexually by two methods. One is called binary fission. One binary fission. So, this is the first type of asexual reproduction in bacteria. One binary fission, and second one formation of endospores. Endospores. So, there are two methods in asexual reproduction. One is called binary fission, second one is called endospores formation. Similarly, this binary fission generally it takes place in this is a common method of asexual reproduction. Common method takes place in favorable conditions. Okay, it takes place in favorable conditions. Very important. Mention the name of asexual reproduction in bacteria that takes place commonly in favorable conditions. That is binary fission. So, binary fission is a common type of asexual reproduction and takes place in favorable conditions. But formation of endospores, this is not a common method. It takes place only in unfavorable conditions. Okay, unfavorable conditions. It takes place in unfavorable conditions. So, the common type of asexual reproduction that takes place in favorable conditions in bacteria, this is binary fission. But rarely some of the bacterial cells in some situations, that is in unfavorable conditions, they also produce endospores. This is takes place in unfavorable conditions. But binary fission it takes place in favorable conditions. So, binary fission is nothing but mitosis. Is nothing but mitosis, where the cell eventually divides into two daughter cells in favorable conditions. So during the time of bionutrition, first the bacteria, it is transversely elongated in favorable conditions. Transversely elongated in the favorable conditions. Now this is represented by cell wall, and cell wall is followed by plasma membrane. This is the bacterial cell. First, bacterial cell, it is just transversely elongates in its size. Then this bacterium is also represented by the infoldings in a plasma membrane. For example, this is a plasma membrane. You know, bacterial cells also contain some small invaginations, means infoldings. The infoldings of plasma membrane means plasma membrane, it is invaginates or it produces infoldings into the cytoplasm. Such regions, they are called mesosomes. They are called mesosomes. The infoldings of this plasma membrane are called mesosomes. Now, these mesosomes, at the region of mesosome, this bacterial cell will show a small invagination on either sides. Okay, here first cell, it elongates in its size. Then it is followed by replication of DNA. Replication of DNA means the bacterial chromosome means the nucleoid present in bacteria. Nucleoid, this means the nucleus without nuclear membrane. That is also called xenophore. First, the xenophore, it divides into two parts. First, the DNA, including the total nucleoid, it divides into two parts. Now these two, they transfer to the opposite poles of the cells. Means this nucleoid first divides into two parts. Now, at the region of the mesosomes, these mesosomes, they grow gradually in centripetal manner. Centripetal manner, like this. For example, this is a bacterial cell and this is a nucleoid. At the region of these mesosomes, they show a small constrictions like this. At the region of mesosomes, they show the small constrictions, a small invaginations. Okay, now these invaginations or constrictions, they show gradual elongation in centripetal manner, resulting they touch at the center. So the small constrictions which appear at the region of mesosomes, now they grow centripetally. Centripetal growth means it takes place from peripheral region to the center. Now, in the formation of a continuous layer between the two nucleoids, resulting the total cytoplasm is divided into two compartments consisting of one nucleoid. Thus, this cytoplasm is divided into two parts due to the centripetal growth of the mesosomes present in a plasma membrane. Now, this cell wall, it is also shows a small constriction at the region of these compartments, between the compartments. A small constriction is also appeared in the cell wall. Now, this constriction, it is gradually deepens like this. This constriction is gradually deepens and resulting the eventual division of parental cell into two daughter cells. This is one of the daughter cell and this is another daughter cell. So here a construction which appears in the cell wall gradually deepens, grows centripetally and touches at the center of the cell, resulting the eventual division of the total cell into daughter cells, two daughter cells. One parental cell divides into two daughter cells. This is nothing but mitosis. At the same time, these type of this is a nucleoid, and this is wall, and this is membrane, and this is a mesosome. Ultimately, one parental cell divides into two daughter cells. In this way, every parental cell divides eventually into two daughter cells. Now, these two daughter cells, they are exactly similar to the parental cells. So, this is the common type of asexual reproduction that takes place in favorable conditions. It's nothing but mitosis, but it is nothing but mitosis, but it differs in some. It differs from the mitosis. How it differs from mitosis? In case of mitosis, okay. In case of mitosis, spindle operation formed. Spindle apparatus is formed in a mitosis. But in case of binary fission, in binary fission, there is no the formation of spindle apparatus. No spindle apparatus. Very, very important. There is no the formation of spindle apparatus in case of binary fission. But this is also a type of mitosis in which every cell, parental cell, divides eventually into two daughter cells. So guys, at the same time, for example, in every binary fission, every bacterial cell, it divides by binary fission within 18 to 20 minutes. 18 to 20 minutes. So, the binary fission it completes within 18 to 20 minutes. In most of the bacterial cells, this time is 20 minutes. So, this time, this is considered as generation time. This is called generation time. This generation time is 20 minutes. In most of the members, it's 20 minutes. Means, in every binary fission, bacterial cells would be doubled. Bacterial cells would be doubled. For example, in or two cells are present in a medium, and how many generations of mitotic divisions that takes place to produce 8 cells from one cell. In first 20 minutes, one is divides into two daughter cells. Okay, in second 20 minutes, two cells are divided into 4 cells, and in one more multiplication, 4 cells are divided into 8. So, every bacterial cell it divides thrice in 1 hour. Every daughter cell, every bacteria divides thrice. Every bacterial cell divides thrice per 1 hour. Per 1 hour. So, in each multiplication, bacterial cells would be doubled in medium. For example, one bacterium is one bacterium it is continued binary fission for 2 hours. One bacterium continued binary fission for 2 hours. What is the total number of cells formed in medium? That is, every bacterial cell it divides for 20 minutes once. Okay, in first 20 minutes, one cell it divides into two daughter cells. Now, in 40 minutes, two cells are multiplied into 4 cells. And in 60 minutes, 4 cells are multiplied into 8 cells. And in 80 minutes, 8 cells are divided into 16. And in 100 minutes, 16 are divided into 32. And in 120 minutes, 32 cells are multiplied into 64 cells. So, how many generations of mitotic divisions are required to form 64 cells from one cell? That is 3 generations in first hour and 3 generations in next hour. That is 6 generations of mitotic divisions are required to form total 64 cells. Okay? At the same time, here how many mitotic divisions are required to form total 64 cells? That is 64 minus 1, that is 63 mitotic divisions are required and 6 generations of mitotic divisions are required. Now coming to another points, this is endospores. So formation of endospores, it takes place in unfavorable conditions. It takes place in unfavorable conditions. The type of asexual reproduction that takes place in unfavorable conditions, that is called such spores are called endospores. Endospores. So these type of endospores, they formed in some specialized bacterial cells only in unfavorable conditions. Here, for example, this is the bacterial cell. This is a nucleoid. This is bacterial cell. During unfavorable conditions, this bacterium, cell wall, it becomes very thick. Cell wall, it becomes very thick, and entire protoplasm, it becomes contraction. Means entire protoplasm, it lose water. Means water move out of the cell. Water, it becomes dry. Now the protoplasm, it becomes shrinks. Now this contracted cytoplasm, it is surrounded by very thick cell wall. This is a cell wall. So this is a nucleoid and this is a cytoplasm. Students, during unfavorable conditions, bacterial cells, they undergo dehydration and protoplasm becomes contracts, and cell wall, when this protoplasm is also secretes a new and a thick cell wall of its own. Now entire cell is covered and protected by very strong thick cell wall. Thus these cells, they are converted into endospores during unfavorable conditions. Now these spores are called endospores. Now these cells, they are considered as endospores. These endospores, they hide over unfavorable conditions. Unfavorable conditions. These spores, they are protected from unfavorable conditions. Means lack of food materials and high temperature and all unfavorable conditions. They are protected, these cells, from unfavorable conditions. After attaining of favorable conditions, each and every spore, it germinates into a new cell. It germinates into the new cell. Such type of asexual reproduction is commonly takes place in unfavorable conditions. Okay, these spores are called endospores. So, asexual reproduction in favorable conditions, it takes place in bacteria by binary fission. But in unfavorable conditions, these bacterial cells, they are also asexually reproduced by the formation of endospores. At the same time, students, very important, arrange the following events in binary fission in correct sequence. 1. Cell, it elongates in suitable medium. 2. Appearance of a small construction. in the plasma membrane. 3. Centripetal growth of this constriction. For separation of the total mean first elongation of the cell. Second one, separation of nucleoid into two nucleoids. And 3, appearance of a constriction in plasma membrane. For centripetal growth of this constriction. 5. Separation of entire protoplasm into two compartments. 6. Appearance of a small constriction in the cell wall. 7. Eventual division of the total parental cell into two daughter cells. So, binary fission, it doubles the cells for every 20 minutes. Such type of asexual reproduction is called binary fission.